Myself, Abhimadhavan, and my teammates Navani and Vishwanathan. Our project title is Extraction and Characterization of Exopolysaccharides from Lactic Acid Bacteria for Food Preservation. Our project is guided by Dr. M. Sivashankari, Assistant Professor, Department of Biotechnology. As concerns over the safety and quality of food products continue to rise, there is a growing interest in natural and sustainable preservatives. So, exopolysaccharide produced by the lactic acid bacteria has emerged as a promising candidate for enhancing food preservation. So, in this study, we aim to explore their potential as a bioactive compound for food preservation through systemic approach and sorting out their physiochemical properties by evaluating their efficiency in inhibiting food spoilage microorganisms. In our project, we have chosen lactic acid bacteria because they are classified under GRAS category, that is generally regarded as safe because so their metabolites are not harmful for a human being. They are without any health risks. We can use their metabolites in a human being. Exopolysaccharides. Exopolysaccharides are high molecular weight extracellular compounds. They are usually sugars. They are usually seen in the outer surface of the cell, forming a loosely attached or slimy layer. These exopolysaccharides can be homopolysaccharides or heteropolysaccharides based on the monosaccharide composition. For example, leaven is a homosaccharide and xanthan is a heteropolysaccharide. Exopolysaccharide plays different role in food industries. Based on the exopolysaccharide, the roles may differ. For example, dextrin plays a role in the improving texture, and xanthan prevents sedimentation, gellan enhances moisture, like that, etc. And these are the predominant sources for the different exopolysaccharides. Our main objective is to isolate and cultivate the lactic acid bacteria from different food sources, and extraction and characterization of the exopolysaccharides. These characterization include functional as well as the structural characterization, and then evaluation of the antimicrobial property of the lab-derived EPS. This is our workflow. At first, we collect food samples like curd, milk, and soy sauce. Then we screen for the exopolysaccharide producing bacteria by which it shows the creamy and slimy layer. So we isolate these colonies and we screen for antimicrobial activity against Bacillus cereus. Bacillus cereus is a foodborne pathogen which is predominantly cause food spoilage. So we have chosen that as the indicator organism here. Then we biochemical characterization the strains here used here, in which we have done indole test, catalase test, and gram staining to identify the bacteria. Next, we extract the exopolysaccharide from the isolates and we structurally and functionally characterize them, in which structural characterization include FTIR and TLC, and functional characterization include antimicrobial assay with the exopolysaccharide and anti-biochemical assays. For isolation of the EPS producing bacteria, first we have serially diluted the food samples, curd, milk, and soy sauce in different dilutions, and we have streaked it up to 6th dilution, and we have isolated of say single colonies and inoculated them in the MRS broth, which is the selective medium for lactic acid bacteria, and we have incubated and preserved the samples. These are the isolates that we have obtained after streaking. FP001 from commercially brought curd, FP002 from homemade curd, FP003 from soy sauce sample, and FP004 from milk sample, in which FP001 shows higher yield of EPS, showing high creamy texture. The tangible outcome from our isolated samples was that we have experimentally observed the antimicrobial activity of the isolates against Bacillus cereus, and the effective samples against Bacillus cereus was exopolysaccharide extracted, and we have done structural characterization of the exopolysaccharide using FTR analysis and TLC, and functional characterization was done for the antimicrobial activity against Bacillus cereus with the extracted exopolysaccharide. We have isolated genomic DNA and amplified the genomic DNA using PCR for 16S rDNA sequencing to identify the species level bacteria. The major impacts with the exopolysaccharide give to the society is by enhancing food safety and being a healthier food option. It also supports for the local food producers in terms of availability and the price, and it is also highly sustainable. Thank you.